My name is Andrew Sawyer from Carl Feng's laboratory in the University of Sydney. Tuberculosis remains a major cause of death and disease worldwide, and a significant barrier in fighting TB is that we don't understand what a successful immune response against the MTB bacteria looks like. At the cellular level, it's clear that the battle against MTB occurs in organised lesions called granulomas, and there is increasing evidence from animal models and human studies that even in the same patient, some granulomas may clear bacteria and others not. So in this study, we aim to define all the types of cellular lesions within the lung tissue of TB patients, as a first step to understand the diverse functions and outcomes of granulomas. We used the OPAL imaging system to stain for major immune populations and used image stitching to capture entire patient tissue sections. We identified individual cells using segmentation, and to capture every cellular lesion, we developed a density-based system that was able to classify lesions into both necrotizing and non-necrotizing granulomas. Overall, we identified 726 individual lesions across 13 patients. When looking at the density of each immune population between different lesions, we saw a lot of variation, but notably less in our necrotizing lesions compared to their non-necrotizing counterparts. Necrotizing lesions had a lower density of both B cells and CD4 T cells when compared to non-necrotizing lesions. Using PCA analysis, we saw that necrotizing lesions tend to cluster together, and that PC1, which was mainly driven by density of B cells. As well as variation in the cellular composition of lesions, we also observed variations in their intra-lesion cellular distribution. While our necrotizing lesions were defined by their central acellular core, we also observed that non-necrotizing lesions could either have a more tight or a more loose, though still cellular, central region. And we developed a metric called Total Cell Preference Index, or TCPI, that compares the density of peripheral and central regions within a lesion, and we were then able to stratify all lesions based on this intra-lesion cellular distribution. Using both our PCA analysis and the TCPI metric, we could then separate lesions into 4 types based on density of B cells and the intra-lesion cellular distribution of all cells. Unexpectedly, we also found that these non-necrotizing lesions appeared to be organized as satellites of necrotizing granulomas. And interestingly, our type 1 and type 2 lesions tended to be twice as close to a necrotizing lesion compared to our type 3 and type 4. And these findings suggest a possible crosstalk between necrotizing and non-necrotizing granulomas. Finally, we used trajectory analysis, a common tool for single-cell analysis, to determine whether there may be a temporal relationship between our lesion types. And the results we found suggest that these different types of lesions may represent different stages of a developmental pathway going towards necrotizing lesions. In summary, we developed a pipeline that can resolve the heterogeneity of all lesions within a tissue. In addition to necrotizing lesions, we also identified 4 types of non-necrotizing lesions. And these, together with necrotizing lesions, form a histopathological superstructure in the TB-infected lung. Overall, this study demonstrates the importance of capturing all lesions within TB-infected tissue to understand the full scope of lesion heterogeneity.